Some of the most popular that you'll hear about at this meeting are genome engineering. So you can use messenger RNAs to express nucleases and transposons. The most popular ones recently have been CRISPR-Cas9, but there's also extensive work and clinical trials going on with zinc finger nucleases, with TALENs, and with transposons such as Sleeping Beauty and Piggyback. Most of these applications are ex vivo. In this case, the reasons to use messenger RNA, again, is that it's transient. So you're causing intentional double-stranded breaks, which would trigger integration of any DNA that's present. And so transient expression may reduce the risk of off-target effects. There's also no risk of insertional mutagenesis there because of that. You have the ability to difficult to transfect non-dividing cells, which are often the targets of these therapies. And you only require delivery of the mRNA to the cytoplasm and not to the nucleus, and so that's another advantage. Another common application is gene replacement. So this is mostly mRNAs delivered in vivo by lipid nanoparticles or polymer delivery vehicles. Again, you want to avoid the risk of DNA insertion because this is now chronic treatment. You can imagine treating these patients for the lifetime. And also some of the target cells are non-dividing cells such as hepatocytes. This type of therapy tends to be liver directed for the most part. And you can fine-tune the expression of that gene by simply changing your dosing during the course of the treatment. Another popular application for mRNA is immunobiology. And this has a number of different areas. So vaccines are very popular. So there are companies like CureVac and BioNTech and Moderna who are using mRNAs for vaccines. This is a platform for personalized medicine. So you can make a personalized mRNA vaccine for a single cancer patient, for example. These types of rapid personalized medicines, though, require rapid manufacturing turnaround times. And mRNA is ideally suited for that kind of a process. And mRNA also acts as its own adjuvant. And there are some literature that suggests that you get activation of different MHC pathways when you express an mRNA intracellularly versus expressing or delivering a protein extracellularly. And so it may have some advantages. Another interesting area here is the area of self-replicating RNA or replicons. These are often based on viruses such as alpha viruses. And you can give a very low dose because the RNA, once it gets inside the cell, goes through replication cycle, although it doesn't produce active virus particles. And by going through a double-stranded intermediate, that activates immune pathways and it acts as an adjuvant. Again, people are interested in these kinds of things for responding to pandemics or bioterror incidents. And so rapid manufacturing times are also critical here. And a last one that I want to touch on is the inactivation of checkpoint inhibitors using CRISPR. It's another popular application of messenger RNA. All right, so now I'd like to spend a little bit of time telling you about how your body recognizes self-RNAs, distinguishes them from non-self RNAs.